Welcome back to the course of chemical crystallography. Today we are in the powder X-ray diffractometer room where we will be showing you how a powder X-ray diffraction data is recorded, how the sample is prepared and how we can process the data using a computer software program. So to start with, I will first show what are the required things for a sample preparation. So what we need is a mortar and pestle. This is agate mortar and pestle which is made up of a stone, and this stone is very hard. So when we grind the sample here, this does not make any scratch on the stone and no sample gets stuck to this stone during the sample preparation process. So once we have powdered a sample using this mortar and pestle, we need to transfer them to these sample plates. So these are the sample plates on which we need to spread our samples. You can see that the central region in these plates are etched, and these etched plates have different depth. So depending on the amount of sample that we have, we should choose a particular sample plate so that we fill the entire groove here with our sample. So these plates come with different depth like 0.1, 0.2 and 0.5 millimeter depth, and the size is about 1.5 centimeter in square. Technically one has to fill the entire square with sample, but in case we do not have enough amount of sample, what we need is we just fill the central zone of the sample plate and place it on the diffractometer. To make the sample, we transfer the crystallized material. It may be nice crystals. So this is a polycrystalline powder and we need to grind it gently to an uniform particle size. This grinding should be done very carefully, and harsh grinding may lead to conversion of the sample to a different form. In case the sample contains some solvent, it may so happen that during this grinding, if we apply too much of pressure, the solvent can evaporate and lead to a different crystal structure than the starting material. So we need to then scoop out this sample. You see that while I am scratching, the surface of this stone is not getting scratched. Since we have very little sample, in this case we need to choose one of these plates which has the lesser depth. So this plate, which I can feel with my finger, has a lesser depth. This is about 0.1 mm depth sample plate. So we should place the sample in the central zone of this plate. We can scratch it further to take the remaining amount of sample that is there inside the mortar and pestle, this mortar. So now what we need to do is to bring the sample to the center, because we do not have large amount of sample. We have made a heap at the center, and then we need to make it flat from the top using a glass plate so that the top surface is flat and the sample is uniformly spread in the center. So now this sample is ready for recording powder X-ray diffraction data. We need to clean the side so that the excess sample is not getting contaminated on the diffractometer. So what we will do on the diffractometer is we will place this sample in front of the X-ray beam, and then we will make the sample rotate like this at 120 rpm so that the entire sample is evenly exposed, because the beam that we have here is a line focus, which means the beam is like a line which is like this, and then if we rotate this sample below the beam, the entire sample gets exposed to that beam. So this removes any special orientation effect that might be there in those microcrystalline particles. So this is the powder X-ray diffractometer which we use to record the powder diffractograms. So when we try to open the door, we press the button and then open the door. What we have here are the equipment with the X-ray tube, detector and the sample stage. On the left hand side here, we have the X-ray tube mounted. This is mounted from top, and you can probably see that this is a copper based X-ray source which is written here as copper. So the tube is giving you copper K alpha and K beta radiation out of it. So when the beam comes out, this is the optics where it is called the cross beam optics. It then makes the beam in a linear fashion, and then we have some slits here. This is a Bragg-Brantano slit. It is written as BB. So the beam passes through that opening zone, making it as a line focus source. Then we have some solar slits here. We have a height limiting switch here which limits the size of the beam to 10 millimeter. As we can see here, it is a 10 millimeter slit. This slit can be made smaller in case of very small amount of sample. This is the sample stage which can be rotated like this. So during the experiment it rotates, which then makes the sample uniformly exposed to the beam, and it removes all possible Sprefford orientation effects. The X-ray beam comes from the left hand side, gets diffracted from the sample placed on this plate here, and then gets diffracted and gets recorded on the detector which is written Rigaku at the back. That is the detector for X-ray here. So before the sample, before the X-ray beam goes to the detector, we have a beam stop here. That beam stopper stops the direct beam from going to the source, going to the detector, because the direct beam is very high intensity and it may spoil the detector, and that is why we should have a beam stop. So in this X-ray tube, we need to cool the anode which is at the back of this tube. So to cool that anode, we have water supply coming through these tubes which are here. So this is the tube through which the hot water is going back, and there is a tube inside here through which the cold water is coming in. So we have a chiller in a different room which cools the water to about 16 to 17 degrees centigrade and then runs it through the tube at about 4 liter per minute flow rate, so that the cold water comes, hits the backside of the anode, takes the heat and goes out to the chiller. This chiller is an important part of any X-ray diffractometer which has to be maintained at the prescribed temperature. For this powder diffractometer, we maintain it at 16 degrees centigrade. When the X-ray data is going on, we will see that this particular red light will be on, which indicates that the shutter is open. When we are keeping the door open, that means the shutter is closed. So although X-ray is generated inside the tube, that X-ray is not coming out to this chamber until and unless the shutter is open. And one cannot open the shutter without closing the door. So when we start recording a data, we will show you that when the shutter is open, this red light will be glowing. That red light, the red light on the top indicates that X-ray is on, which means the tube is on and it is producing X-ray. So in case there is a red light on that, it indicates that the X-ray is on. If there is any failure and the generator has stopped working, that red light will be switched off. Here in this chamber, once again we have some solar slits, and what we have before the solar slit is a nickel filter. This nickel filter is used to remove the K beta component of the copper radiation, and what reaches the detector is just the copper K alpha. So this diffractometer works in a theta theta geometry. So that means here the X-ray source and the detector will go simultaneously upwards. In this case, this works on a theta theta geometry. So I have the X-ray beam coming from my right hand side and the detector is on my left hand side, and during the measurement both the detector and the source goes up slowly. So this is called the theta theta geometry. The other geometry that is possible is a theta to theta geometry, where the source is kept fixed, both the sample and the detector moves upwards, and that is a different geometry possible for X-ray diffraction, powder X-ray diffraction measurement. So now I am going to place the sample on the diffractometer. We need to remove this dummy plate, which is another sample plate. We now would place the sample here. So this sample is now placed in the middle portion. If we see that here at this point, the X-ray beam will come and hit. As a result, if we rotate the sample during the measurement, it will expose the entire sample to the beam. So we need to close the door because we cannot run X-ray diffractometers with the doors open. For safety reasons, we should not be exposed to these X-rays during the experiment. So when we close the door, we are safe to operate the instrument and the door is locked. So what happens inside is X-ray beam comes from left, gets diffracted on the sample and gets recorded, and in the front we have a glass door through which we can observe or monitor the process of this data recording. So in this window we can see that the X-ray is on and we are operating it at 40 kilovolt and 40 milliamps of power, which is equivalent to 1.6 kilowatt. So now we need to set up the data collection strategy. As we normally do, we record the powder X-ray diffraction data from 3 degree to about 50 degree into theta. This is a sampling width, which means at the interval of 0.02 degree there will be a data recorded, there will be a point, and we need to decide on the scan speed of data collection. In general, we do it for 5 or 10 degree per minute, but in case we have a small amount of sample, we do it at a slower rate. So normally the slower rate can be about 1 or 2 degree per minute. So for today's sample, I am going to do it for 5 degree per minute scan speed. Change the current here to 40 milliamps, and these are the default settings of different slits that I have shown on the diffractometer, and as long as we don't change the slits, we don't change any of these numbers. So this is the window where we set up the data collection strategy, and from here we ascertain that the sample will be rotated by 120 rpm during the data collection. That means the sample will rotate in an horizontal direction at 120 rpm during the entire span of data collection. So now we need to give a name of our sample. So we can give it a name as test 2.2 and say it test 2. And then on clicking this yellow button, it will start executing the measurement and it will show how the pattern is coming. So here you can see that during the measurement, the sample is rotating at 120 rpm on the sample stage, and the data is getting recorded on the computer, which is going to be shown soon. So now we are going to analyze the data using the software available with this diffractometer. This software is called PDXL. So we are going to open the raw file in this software. So what here is done is that it has opened the powder X-ray diffraction data that we have just recorded, and we have a few tools to examine the data. If we try to see the background, how it is, we only need to expand that region and we see that the background is not really flat, but the background is wavy in nature. So this wavy background indicates probably the sample is not fully dry, it may be having some solvent in it, and it is not a very good sample for powder X-ray measurement purposes. What we can see here is that, of course, we have sharp peaks. As we can see, if we expand this region, one can see the peaks are single. So the red ones are observed and blue ones are calculated peaks in this pattern. And this particular software has already found out the correct positions of those peaks and has the software has already identified those peaks and marked them as blue sticks for indexing purpose. So one can also do a powder pattern indexing using this software. If we see the mid-angle region, what we see is that the peaks are very broad and they are not well resolved. So this is the problem of a home-based source which cannot resolve the peaks which are very close to each other, and that is why we sometimes try to record powder X-ray diffraction pattern using synchrotron sources. The advantage of synchrotron data will be that it will be highly resolved. These peaks will have very high intensity, and we should be able to solve and refine the structure from this powder X-ray diffraction data. Now what we have done is that we have removed the background, and what we are getting are the alpha 1 and alpha 2 peak positions that we can see. In this case, if we fix that particular point as alpha 1, then the green line which is coming next to it is the peak for K alpha 2. So this particular powder X-ray diffractometer does not use monochromatic radiation, but uses just copper K alpha 1 and alpha 2 as a mixture, and hence the peak finding software indicates the possible positions for K alpha 1 and K alpha 2 as well. This can be also seen at high angle peaks. So, the alpha 1 alpha 2 split is there wider at the high angles compared to the low angles. If you can see the red line and the green line, they are well separated at higher angles like 30°. If we go to a further high angle, something like 48-49°, you can see that the alpha 1 alpha 2 split is much larger, but for this low angle peak they are extremely close. This is the characteristic feature of powder X-ray diffraction when recorded using two different wavelengths. So, if we had not put the K beta filter, we would have got another peak corresponding to K beta radiation, and the pattern would become more messy. So, here in the screen what we are now seeing are two diffraction patterns. One is recorded in red, one is displayed as red, the other one is displayed as blue. The red one is with a nickel filter installed before the detector, and the blue one is without the nickel filter placed in front of the diffracted beam. So, what we see in general in the blue pattern is that there are some additional peaks which are not there in the red. That indicates the additional peaks that are coming are because of the presence of K beta radiation in the diffracted beam. That presence of K beta radiation is not very prominent for the weak diffraction peaks, but still we can see that if we place the K alpha 1 on this line, there is a K beta peak coming here which was not there in red. Similarly, if we bring that line here on the right hand side, this peak corresponds to the K alpha 1 and then K alpha 2 as well, but there is a very small peak which is appearing there, which was not there here because it was a flat background, but here there is some indication of small peak. If we go and zoom near the highest intensity peak, it becomes very much evident that if I make that peak as K alpha 1 and the alpha 2 is immediately next to it, this blue peak which has appeared in the blue pattern is the K beta peak which was missing when we used the filter. This case becomes more significant in the high angle region where we have lots of peaks and some peaks which are actual peak from the sample peak for K alpha gets merged with a peak of K beta of another peak and makes the powder X-ray diffraction pattern broad. So, if I try to look at this particular peak which is here and I then focus it as K alpha 1, the corresponding K beta appears here, but you see in the red also there is a peak. So, this original peak is now merged with K beta and become broad. The next peak that we have in the main pattern is now focused here. So, this peak is the K alpha 1, but the corresponding K beta peak is here which was not there in the original pattern. The next larger peak which is somewhere here is the K alpha peak. The corresponding K beta appears at much lower 2 theta value and as an independent peak which was not there in the red. So, this pattern comparison clearly shows that if we have forgotten to put the K beta filter, that is the nickel filter for copper radiation, we will get additional peaks. And then if we throw these peaks without knowing that they are from K beta, any indexing software will fail to index this powder pattern. So, the presence of K beta filter in all the diffractometer is essential, and we must not forget to put that. The K beta peaks are both here shown for these two peaks, which are here in about 25 degree into theta. And this spread keeps on increasing at higher and higher angle if we try to see it at high angle. And in most of the cases, then that this is the peak for K alpha, the K beta peak appears somewhere there, which is merged with one of those actual peaks, and we don't get a different peak for that. See here again, this peak is the K alpha peak, the corresponding K beta is here. As a result, this peak intensity has increased. So, K beta is now hiding behind a genuine K alpha 1 peak. And since we recorded this data only up to 50 degree to theta, we have peaks up to about 46-47 degrees. So, this particular peak also once again appeared somewhere there as a broad, which was not there in the red in the original. So, this shows that the presence of K beta becomes more and more significant for all kinds of peaks, small angle to high angle, very weak reflections to strong reflection, and hence it will then create problem in data indexing. So, always we must remove K beta either at the end of the source, that is before the diffraction occurs, or before the diffracted beam meets the detector and generates signal, the K beta filter has to be placed. K beta filter can be placed immediately after the optics, before the X-ray falls on the sample, or after the diffraction occurs from the sample and before the beam reaches the detector. Here we are trying to compare the powder X-ray diffraction patterns of two polymers. As we have already discussed in the class, polymers are different crystal structures of a particular compound. That means the internal structure of the lattice is different. As a result, the unit cell dimensions could be different, and hence the peak positions in powder X-ray diffraction data will be different for different polymers. So, here what we can see is that there are two powder patterns, red and green. These are recorded on two different polymers of same compound. You see the peak positions are different. In some case, the peak is shifted towards left. At this region, the peaks are totally different because you had 1, 2, 3, 4 and 5 peaks, while you have only 1 and 2 green colored peaks. So, this type of difference in powder X-ray diffraction pattern clearly indicates the polymorphism present in this particular sample. Of course, if the sample is completely different, if the two patterns, then the two patterns can be different, they are not polymorphs. So, we need to have a prior information about the origin of the sample. If we know that the sample is containing a particular compound and it was crystallized in two or more different methods from different solvents and so on, and we have resulted a powder material originating from the same compound, 4 or 5 different crystallization methods yielding differently looking crystals, then the powdered X-ray diffraction pattern has to be recorded on all the samples, all the powders, and then compared like this by peak to peak matching, to see whether there is a difference in powder pattern as we are seeing here. This particular case where the sample we know has originated from one particular compound and is giving two different crystal structures with two different sets of peaks appearing at two different points. So, this is how one can identify polymorphs using powder X-ray diffraction data.